or discover them through any kind of method, this is the path that we want to take forward. So I would say there's not much of a difference.
Do you consider modulating post-translational modifications as a way to, regulate PPIs like phosphorylation, kinases, that type of thing? Does that fit into this strategy?
So modulating a kinases?
Well, given that post-translational modifications are very important towards protein-protein interactions, can you consider to modulate those post-translational modifications in specific ways? If you want to increase phosphorylation of a particular protein, to increase its association or decrease it, or acetylation or something else.
Well, in this case, do you think you really want to really to interact with the formation of the protein complex? It's probably not the case anymore, right?
I'm thinking more along the lines of something, for example, let's say NF-kappa B, the ability of NF-kappa B subunits to interact with other proteins is controlled by phosphorylation, acetylation. So if you could selectively design tools to modify those sites, either increasing or decreasing, that would be a way to modify those protein interactions.
Okay, so I see in this specific example, if it would be a phosphorylation, one would think about finding an agent that would bind to the site that will be phosphorylated and block this site from any kind of phosphorylation.
Right.
So this is certainly a possible approach to look into that has not really been pursued so far at this time, right. But it goes through the same principle, is understanding what are the key amino acids in this region that we could use to right. develop agents against them.
I think there might be, I want to say on the order of 200 or so different types of post-translational modifications that have been described. And so those must offer some unique opportunities for modifying interactions.
Yeah, I think it's a very good point, and it's one area that we have to look into as additional potential targets.
So on one of your slides, you mentioned that the predicted size for these types of molecules would be about 600 to 700, potentially higher, right? So I'm curious, if one looks at historical data, how much more difficult or how much less likely is it to succeed and reach the drug stage, and in particular how that impacts drugs in the context of CNS space when one has to consider the blood-brain penetration barrier issues?
Yeah, I think this is a very good question, and you're right. First, the prediction are we're going to need larger molecules if we want to interact with protein-protein interactions. So we've seen that the example I gave was for oncology, where the compound is already bioavailable, is cell penetrant because BCL2 is within the cell. So it has been possible to advance such compound for oncology. Now for CNS indication, this is a very different story, and maybe I didn't stress it out enough in the presentation. But now we have to make sure that those compounds are going to be brain penetrant. So how are we going to do that with molecule of that size? So we don't really know at this point. But if we look at historical data, we know that the mole weight has to be below 500, probably even lower, to have a good brain penetration. So we don't really know how to do that. So we, in parallel to discovering those compounds, I think it's going to be very important for us to have better drug delivery technologies. An example has been, it's not really drug delivery technology, but in the area of antibodies, for example, recently Genentech developed a bifunctional antibody. So one arm of the antibody really recognizes the protein, while the other arm is used to help this antibody enter the blood-brain barrier. And they took advantage of transport proteins in the brain, like proteins of the transferrin classes. And apparently this specific antibody is now brain penetrant and very well distributed. So for small molecule, a molecule in this size, 700, 800, we might think about those type of technologies. This is really the future, but we have to this is going to be very difficult for us to do that.
Thank you.
Thank you.
Our next talk is from Dr. Ravi Krumpel from Pfizer, who's going to be discussing kinases as a molecular target.
So while they're setting up the computer, I also want to thank Mark and Howard for the invitation to be here. I've thoroughly enjoyed the discussion and presentation yesterday, and look forward to the other talks today. All right. It's interesting. Mark said something about untouchable target. For some people, GSK3 beta might be an untouchable protein kinase. We'll see what the data that we have. Before I go further, I wanted to quickly acknowledge the large number of colleagues at Pfizer who contributed to the latest round of the GSK3 beta project. We worked on it. I think the industry has worked on this target for a good 15 years now, starting from diabetes. And I'm just going to describe to you some of the work that we did in the last iteration. So here's a quick overview of my talk. I'll give you a little bit of background on GSK3 beta and its role in Alzheimer's disease, and some of our efforts in developing GSK3 beta inhibitors. And Howard especially wanted me to address or talk about the property space. We heard quite a bit about it yesterday from Laura and Heather, but I'd like to maybe focus a little bit more specifically on the property space for protein kinase inhibitors and see what are the opportunities and challenges in this area. And then I will address something about the kinase selectivity. And I see that I think there was going from one computer to the other, something has been messed up, so pardon me. And then some of the work that we did in assessing the safety of target cellular safety of the GSK3 beta as a target. I think we had a lot of discussion about target validation, and I will hopefully walk you through some of the work that we did to validate or invalidate GSK3 beta as a target. And then I'll end with conclusions. Protein kinases, as we have, I think most of you have, I'm sure, very familiar with. It constitutes about 2% of the human genome. And as we just heard, plays a key role in post-translational modification. And it's involved virtually in every signal transduction cascade protein kinase is involved. And there has been a lot of interest, therapeutic interest in protein kinases. It's implicated in disease, various disease pathologies. And they are, I wanted to say, they are attractive targets for intervention because of the property of the ATP site, which is what we have targeted, we and others have targeted primarily for protein kinases. It presents a good mix of hydrogen bonding properties as well as lipophilic interactions that you can easily target with heterocycles with appropriate functionalities. And because of our corporate collection, not only in Pfizer but also in virtually all the other big pharma, you do get a good attractive chemical matter as starting points. And there are some issues with protein kinases, lack of efficacy that could happen because of questions that came up quite a bit yesterday of targeting a single molecular target. We've had cases where we have advanced compounds to phase 2 clinical trials where we can inhibit a certain target virtually 100% of the target. However, I think what we find is that the signaling can go on through alternate redundant pathways. So that could raise issues for efficacy. And then obviously if you don't have specificity, you could have tox issues. So, you know, my slides are completely messed up going from. Is there a way we could deal with this? Excuse me. Sorry about that. All right, that looks a lot better. GSK3 beta, quickly, it is a serine threonine kinase. There are two close members, alpha and beta, about 98% identical. And those of you who are not familiar with protein kinase crystal structure, that shows the ATP site where one of the substrate ATP binds. That arrow shows to the other site where protein substrate bind. And then GSK3 beta also, in addition, has other binding sites for proteins such as axin and others. There are two interesting things about GSK3 beta. One of them that it is a constitutively active enzyme. It's always in an on stage. It's already on. And then it is because of signaling either through the insulin or the Wnt pathways, it gets turned off. So it's an interesting case. And the other interesting aspect about GSK3 beta is that it requires priming of substrate, a prior phosphorylation of protein substrates by potentially other kinases, and then it has optimal activity for such substrates. The role of GSK3 beta in Alzheimer's disease, we often talk in our industry as confidence in rationale or confidence in mechanism. GSK3 beta has been implicated in multiple pathways in neurodegeneration. So let me walk you through one of them. Microtubules, which are considered as the cellular highways for trafficking, is usually associated with a protein called tau. And tau has anywhere from 25 to 40 different phosphorylation sites on it. Some of those phosphorylation sites are absolutely required to keep the integrity of the microtubules. And quite often, a number of kinases, including GSK3 beta, CDK5, MARC, and many other kinases, they are known to hyperphosphorylate tau. And when you hyperphosphorylate tau, two things happen. One is that the microtubules get destabilized, so you can have issues with synaptic trafficking and other issues. The other possibility is that the hyperphosphorylated tau can polymerize itself and lead to neurofibrillary tangles. Both have been implicated as causative agents in Alzheimer's disease. And finally, in the signaling cascade that's involved in A-beta-mediated neuronal death and apoptosis, GSK3 beta is a player in that signaling. So, there are many evidences that suggest that this plays a role. The expression and the activity of GSK3 beta is upregulated in the hippocampus, and overexpression in transgenic models, mice models, animal models, lead to NFT and neurodegeneration. What is the major issue with GSK3 beta as a target? So this comes back to what Mark said. Is this an untouchable target? So the major issue for us and others has been the confidence in the safety. And why is there a problem with GSK3 beta as a target? So it turns out that it plays a major role in another signaling cascade called Wnt signaling. Wnt signaling is critical for development and for normal physiology. And when the Wnt ligands bind to extracellular receptor, something gets completely, decomposed. So there is a complex called axin complex that has axin, APC, beta-catenin, GSK3 beta, CK1. This complex gets disrupted when Wnt ligands bind, and as a consequence, beta-catenin gets released into the cytoplasm. Typically, beta-catenin is phosphorylated by GSK3 beta, and that keeps beta-catenin, that actually it leads to proteasomal degradation of beta-catenin. However, when that complex, the axin complex, it decomposes, then what happens is the beta-catenin it translocates into the nucleus, associated with transcription factors. That leads to gene transcription. So the concern is that this could lead to cell proliferation. And I think it's pretty clear that if you inhibit GSK3 beta 90 to 100%, you will cause cell proliferation. And what is never known is how much of inhibition of GSK3 beta is required for pharmacological efficacy. There are reports that suggest that you could get away with as little as 25% inhibition. But that point, I think in the context of some of the discussion that we had, it's very difficult to assess going from a transgenic mice model or some animal models to humans. So the question that we had was, will there be sort of sufficient therapeutic index for GSK3 beta inhibitors? And we tried to assess this in a number of different ways, which I'll show. So, some of the key challenges for us is developing kinase inhibitors that penetrate the blood-brain barrier. I think there was a lot of discussion about this. People have often thought that the chemical properties that you need to inhibit a protein kinase are almost orthogonal to the properties that you need for brain penetration. So this might be inherently a challenge. And obviously, there is always the issue of safety. And for GSK3 beta specifically, we also wanted to know whether the target itself is worth it. So here is a quick look at all of the kinase inhibitors that have been approved to date. There are about 16 protein kinase inhibitors that have been approved to date. And those shown in blue are the ones that discovered by Pfizer, actually, and 3 of them we are marketing it. One of them we had to out-license during one of the acquisition process. And one thing that I wanted to point out is that most of these kinase inhibitors, if not all, actually exhibit their pharmacological efficacy through inhibition of multi-kinases. I think you can tell, for example, Sutent, which has been approved for multiple indications, Sutent hits a variety of different protein kinases. When we looked at the property space, I think Laura and Heather talked about quite a bit yesterday, so I won't repeat all of it. But we also had done very similar analysis within Pfizer, and what we found was that certainly molecular weight, polar surface area, and C log P or log D, which is another variant of C log P, are important. But what we also found was that number of hydrogen bond donors was absolutely critical. For example, if you look at this, what we call as a spider plot, the median value for the number of hydrogen bonding donors for CNS drugs is actually 1. And there was virtually no drug in the class that we analyzed with the prescription greater than a certain number in 2004. Not a single molecule had more than 2 hydrogen bonding donors. That actually tells you some of the difficulties in brain penetration if you have a large number of hydrogen bonding donors. This could be mostly because of issues related to effluxing out of the CNS by the P-glycoprotein. So based on this analysis, Pfizer scientists, many of my colleagues, went out and designed a score. We call it CNS multi-parameter optimization or desirability score, combining C log P, log D, hydrogen bonding donors, polar surface area, molecular weight, and pKa, which is what we thought are the more critical part for CNS penetration. And I wanted to quickly walk through a publication by Travis Wager and others. So the way we went about it is that rather than coming up with hard cutoff, for example, looking at C log P, we assigned a score of 1 as long as the C log P is less than 3. And if the C log P is greater than 5, it gets a score of 0. And anything in between, it gets some kind of a graded score. And so similarly, we did that for all the other 5 properties. And for each property, there was a score. And I think most of them had a step function, except for polar surface area, where we had a hump function. Anything less than 40 was, between 20 and 40, it had a graded score. And between 90 and 120, it had a graded score. And anything over 120 got a score of 0. And, so every compound that we synthesize for a CNS program in Pfizer, whether it's for protein kinases or for any CNS program, before we synthesize, we put it through a computational algorithm to predict what the MPO score would be. And there would be very few compounds we will make if the score is less than 4. And I think it's all a question. This is not a rule. It's just a guideline, just trying to help us get into a better property space. And here are 2 examples of compounds that we had taken into clinic. In one case, the log D was less than desirable, and the other, there were 2 properties that were not great, but overall, we still had a reasonable MPO score. I'm not going to spend a lot of time on this slide. What I did was I actually took the entire list of the approved kinase drugs, and we put it through our computational algorithm with the help of Paul Galatsis, one of my CNS medicinal chemist colleague. And we calculated all of the properties and calculated the MPO score. On the very last column is the calculated brain availability. And what you can tell is that many of the kinase drugs that are in the market, they have, when it comes to hydrogen bonding donors, there are quite a few of them. And this is primarily because of what you are required, what the protein kinase ATP site requires for you to bind. And so naturally, their MPO score is low, the calculated brain availability is low. There are a few drugs that are approved that have predicted MPO score greater than 4 and predicted brain availability in a pretty good range, especially the last one that got approved in November of last year, Jakafi, JAK1/2 inhibitor from Incyte and Novartis, has excellent calculated brain availability. I don't quite know whether experimentally this has been validated or not. The other point I do wanted to quickly go over is on selectivity, the issue of kinase selectivity for CNS compounds. One thing is, especially for compounds that bind at the ATP site, we always see a right shift in potency when you go from enzyme assay to cellular assay. This is because in the enzyme assay we use an ATP concentration that's at the Km value for ATP, which tends to be sometimes 5 micromolar, sometimes 50 micromolar, whereas the ATP concentration in the cell is somewhere in the 1 to 2 millimolar range. So you can actually theoretically calculate what the expected shift would be. And however, these shift, they are not the same for different kinases because it all depends on the Km of each of the kinases. So for those kinases that have a Km, like GSK3 beta, which has got a Km of ATP about 5 micromolar, you expect a much larger shift. Whereas other kinases which have a Km for ATP in the hundreds of micromolar, for example, one of the targets we have is called IRAK4. It's got a Km for ATP about 600 micromolar. You don't expect a whole, big, large shift. And so that could lead to potentially unanticipated off-target activities. So that's something to keep in mind. And the other thing that I do want to stress here is that one consequence of limited brain availability of the compound is, if you wanted to think in this way, is the peripheral concentration of the drug is much higher compared to the brain concentration. Let's say that the brain availability is 0.3. It means that you have 3 times higher concentration in the periphery compared to in the CNS. And this could potentially lead to either on-target toxicity if you hit a target in the periphery that you don't want to hit, the same target in the periphery, or it could potentially lead to off-target toxicity. So something to keep in mind. So it's at least my thesis that the selectivity for every kinase inhibitor, you certainly want to address the selectivity is a question, but this may be more of a question more of an issue for CNS kinase inhibitors. So I think I'm almost running out of the time, but I wanted to quickly walk through some of the data that we have for GSK3 beta inhib program. So here is a compound, the optimized compound that we have, about 2 nanomolar in the enzyme assay, and in the whole cell assay, it's about 450. So a 200-fold shift going from enzyme to cellular assay. We show good inhibition of phospho-tau, 60 to 80%. I'll show the data. Inhibition of glycogen synthase in the periphery. And the property, C log P, is in a good range, good solubility, no efflux issues. Experimentally, we measure using an assay where we can measure the compound. The clearance for predicted clearance for human microsome is reasonable, and the brain to plasma ratio is somewhere between 0.5 to 1. And in all the safety assays that we did, whether it's a cell viability or cardiac safety assays, some gene tox that we heard yesterday, or broad screen panels, we had very good, the compound is safe. The issue was that in rats, we do have high clearance, and then we do have some P450 interaction potentially mediated through the triazole of the compound. In terms of property, very good ligand efficiency and lipophilic efficiency, and a good MPO score. Looking at the crystal structure of the compound, it binds at the hinge region of the ATP of the kinase, N-terminal lobe here, C-terminal lobe here. It forms 2 hydrogen bonds, and then we have a good pi-cation interaction mediated by the triazole with an arginine. Looking at the kinase selectivity, we profiled this compound against half of the kinome, 250 kinases in functional assay, in Vitrogen functional assay, and we had 1,000-fold selectivity against every kinase that we tested. And we also tested it in a binding assay at ambit against 3/4 of the kinome, and it's one of the most selective compounds that we ever, anyone has reported. And here is showing the pharmacological efficacy. We can measure the compound either in the brain, or in the brain is shown in purple, and in red is shown in the periphery, in the glycogen synthase. We see about 80 to 95% inhibition at 50 mpk sub-Q, and we see a dose-dependent effect. And the important thing that I wanted you to look at it is that these are the experimentally measured free drug concentration at different doses. So we are getting at 30 to 50 mpk, we're getting somewhere around 3 to 5 micromolar free concentration of the drug in the brain, in the CNS. And the total concentration would be 10 times that value because of the protein binding. So we are able to get this compound into the brain at least 8 to 10 micromolar in the CNS. So, just this concept that kinase inhibitors will not get into the brain is, I think, it's certainly something that you wanted to take with some grain of salt. And here is showing you data of the phospho-tau inhibition versus the free concentration. You can essentially draw a straight line, and we have most of these data has been generated with rat, but we also put it in a transgenic TG4510 mice model and show similar efficacy. And there's a very good correlation between PK and PD. Here's the exposure as measured experimentally, and here's the phospho-tau inhibition and the single dose PK. How much time do I have, Mark? Am I out? All right. Okay, so let me quickly get to this data point. I think I told you about a potential safety issue is that you could actually lead to accumulation of beta-catenin in the cytoplasm, which can then translocate into the nucleus and can cause potentially proliferation. So we measured beta-catenin translocation in HeLa cells or U2OS cell lines, and typically we saw that compared to some other GSK3 beta inhibitors, this is one of the weaker ones. And we also looked at gene transcription using a top flash reporter assay. Clearly, compared to some of the other potent compounds that we had, this is about a much weaker compound. And then we looked at the proliferation, either using Ki67 as a marker or BRDU incorporation, and we didn't see as much. We certainly see some proliferation. So the next one shows you the ratio that we get. The EC50 or the IC50 for beta-catenin translocation is about 13x right-shifted relative to the cellular potency, and in this cell model, only about 6.5-fold. But in the gene transcription, about a 46x window, and in the proliferation assay, about 20x window. But I will say that to our safety science colleagues within Pfizer, this window was not good enough. Because if we are going to go into Alzheimer's disease model, they wanted a squeaky clean compound. So we will not take any compounds into clinical trials unless we have well over a 20x window. So we actually did it in a 10-day toxicology study. We took it into what we call as exploratory tox studies. And at the highest dose that we tested, up to 250 milligrams per kilogram, we do see some proliferation in liver or kidney. But the animals survived without any problem. But when we did the histopathology, we could see some proliferation. So, in light of this program was not advanced. So that is most to do, I would say it is much more to do with the target itself than with the compound. What I do wanted to say is that I wanted to end with that. CNS penetrant protein kinase inhibitors, we have actually developed at least in 4 different programs. We have been successfully able to demonstrate selective kinase inhibitors and show brain availability of these compounds using experimental models. So that's all I have. Thank you.
Fascinating target, and I've worked with companies that have worked on this target before as well, and there is a lot of face value to this target in the disease. But when we look at this target, we're looking at one of its activities in order to validate the concept. How is Pfizer planning on dealing with the broad network effects that inhibiting a pivotal kinase will have, not just on tau and Alzheimer's pathology, but on the massive network interactions that it's involved in cellularly?
Yeah. Very good question. I think you could potentially, in terms of experimental models, you could potentially look at a few of the network effect using experimental assays that we have access to it, and I would say that the other way we monitor that effect is by looking at how different animal models tolerate these inhibitors at very high concentration. As high as in one of the kinase programs, we actually ended up testing at 500 milligrams per kilogram, whereas the predicted human dose was less than 100 milligrams.
That only looks at off-target activities. I'm talking about on-target activities at efficacious doses. How does the company feel about taking a frail elderly population and subjecting them to systemic effective doses of a pivotal kinase inhibitor?
I don't quite the way I would think about it is that whether it is a kinase program or a non-kinase program, the question is the same. When you looking at the population, whether it is a GPCR inhibitor, whether it is a kinase inhibitor, there is no difference there. And so the paradigm that we have been using, the industry has been using all along, and that is at several multiple of the efficacious concentration of the compound, you monitor the toxicity effect in animal models. And yesterday, John Sagers went through a thorough safety evaluation. Very similar to that, we go through and we do that. And if 50 to 100 multiples of the exposure that you need, you don't see a toxicity, it gives you confidence that perhaps that could translate into humans as well.
I just had kind of a general comment. I enjoyed your talk. So I don't know what percentage of the audience is medicinal chemists, but I imagine that there's maybe more neurobiologists or biologists here than medicinal chemists. And this Lipinski rule has been drilled for a couple of days, so I just need to say something on it. Okay? He did a really nice job with this CNS MPO, right? The molecular properties for developing therapeutics for the CNS are, first of all, these are all guidelines. There's a case-by-case basis. You should consider everything in its own context. But the molecular properties that he put up there for TP polar surface area in between 25 and 75, and C log P around 3, and the CNS MPO, the paradigm that you had up there is a good model for looking or for developing CNS therapeutics. I imagine if you didn't know anything about drug design and you came here, you're going to leave with the idea that the Lipinski rule, everyone's talked about it, and it should be talked about in the broad context of drug development, not specifically for the CNS. And we've seen it a lot, and I would encourage anyone who actually is serious about CNS drug development to look at the parameters that they're looking at in their molecules and try and install that into your own molecules.
So the next speaker is Sean Stauffer from Vanderbilt University from PR panel.
Okay, thank you. It's a pleasure to be a part of this conference, and it's been a stimulating 2 days now. And actually, I'd actually wanted to digress somewhat from the topic I'm going to discuss today in line